All right. Hello, everyone. It's a great honor to be here and to talk to you about one of my favorite topics ever. My name is Elaine Hsiao. I'm a professor here at UCLA, and I'm director of the UCLA Goodman Luskin Microbiome Center. Joining me today are two leaders in the microbiome space. We've got Dr. Greg Donaldson and Dr. Chang, and I'll go a little bit more into detail about their expertise in a second. To start, I think that we oftentimes think of ourselves as individuals, but each of us is really a walking ecosystem. We all carry hundreds of trillions of microbes that are in and on us that are serving more and more like a hidden organ. And researchers are finding that they affect everything from our digestion to our immunity and even our moods. Here at UCLA, we feel that we're really well positioned to decode this organ because we have really world-class basic science and clinical care all at the same campus and right next to each other. I think that it's really this unique environment of people who are dedicated on all fronts, on the microbiology, the technology and engineering, and the immunology, and clinical wisdom that's bringing, that's really exciting in terms of bringing the potential to bring new discoveries from the bench to the bedside. We're also really excited about the future, especially the new partnerships that we're building, the team-based science that we're building in terms of our culture, and also our proximity to the UCLA Research Park and the California Institute of Immunology and Immunotherapy down the road is bringing a lot of promise, this field of dreams, so to speak, in terms of interdisciplinary collaboration. And so I'd like to share with me, join me in this discussion, are two experts here that are really kind of representing this whole spectrum, from the molecular mechanisms to the holistic treatment of patients. So I'll start with Greg first. Greg is a new powerhouse of our center. He's an assistant professor in the Division of Digestive Diseases and in the Department of Microbiology, Immunology, and Molecular Genetics. His work really focuses on the spatial structure of the microbiome and how exactly microbes colonize the gut. Essentially, he studies how microbes decide to live exactly where they live and why that location really matters or why that decision matters. I also have Chang here. Lynn is a world-renowned gastroenterologist and an expert on IBS, irritable bowel syndrome, and the influences of stress on that. She's also the director of the new Walter and Shirley Wong Center of Integrative Digestive Health, which is really serving to look at the whole patient rather than their individual gut symptoms. And so thank you both for joining me. I'd first like to start with you, Greg. I think this field in microbiome, everybody has heard about it, I think, by now. And it really started with advances in sequencing technology that have allowed people to really easily and cheaply profile who's there and in what context. But your work is really showing that exactly where they are matters just as much. So could you tell us more about spatial location of the microbiome and why it matters for risk for diseases like colon cancer from your work?
Sure. Thanks, Elaine, for the introduction, and it's really cool to be here at this event with all these people with really different expertise. As Elaine said, I'm really a microbiologist, so I've been studying bacteria my whole career, and came at this angle of which bacteria will actually colonize you from that bacterial perspective. And so, as Elaine said, sequencing technologies sort of revolutionized our understanding of the diversity of the microbiome and made all these correlations with diseases. But what my own research started to reveal is that there were certain bacteria that can get really close to your epithelial lining, which is the surface of your gut. It's not just any bacteria that do this, and it turns out that the types of bacteria that tend to get really close to you, your tissue, are also bacteria that tend to stick around with you for a long time over the course of your life. So we now know from these longitudinal studies looking at microbiomes, some of the species you have in your gut can persist for decades, maybe your whole life. We just don't have that data yet. And actually, some of these microbes you can even pass on if you have children, or you can share with people that you cohabitate with. But it's not just a random collection of bacteria that you're exposed to in the environment. Rather, these bacteria, their only habitat on Earth is the mammalian gut. And so what that tells us is that there's a coevolution between mammals, animals, and these microbes that only live in our intestines. So we're studying how these commensal bacteria can form a long-term relationship with their host. And so this, you can reveal this if you do like new spatial imaging techniques to look further up in the intestine and find that there's certain microorganisms getting into the mucus, certain microorganisms getting within the crypts, which are these small structures in the gut. And all of this, we think, gives these particular bacteria greater opportunity to impact your health because they can engage more deeply with your immune system, other aspects of health that I think we'll get to. So that's sort of the starting point, I think, of where research is pointing us towards not just which bacteria you have, but specifically which bacteria you have that are in close association with you and might have a long-term relationship.
Nice. And I guess this is really exciting that the advances in the basic science are really moving. From the clinical research side, there's huge promise in the microbiome and interest in using microbes and new generations of probiotics effectively to treat disease. But it seems that research has really relied on stool samples as a window to the microbiome. And if their spatial location is so important, then how do we actually look at this in humans? And how would this knowledge impact clinical practice, Lynn?
First of all, I just want to say I have a hoarse voice for the last 2 days, so please excuse me. Stool is so much more easily accessible, but it's really more of a downstream collection and reflecting on the luminal bacteria rather than the mucosa that Greg was talking about. And then we have the gut, the stomach, small intestine, and colon, and we can access the stomach and the colon pretty easily with endoscopy. The problem is really the small intestine, which is 20 to 23 feet long, which is really massive. And it's not a really easy way to access. So one thing that we're doing here at UCLA with the Luskin Microbiome Center is that we're fortunate to have a lot of great clinician endoscopists, and there is an endoscopist, Otis Stephen, who can traverse the entire small bowel with an endoscope. And we're working together to collect tissue biopsies and fluid at the same region, and he knows where he is because he's directly looking at it and going through the small bowel. And then we build to sequence and compare spatially these microbes, because you really want to know not so much is it a good or bad bacteria, but where is it located, what's its function at that part of the gut, and how is the host or the human responding to that? Because it makes sense that the microbiome is going to be different throughout the GI tract because there's different functions. The stomach and the small bowel and the colon all have a different function, so it makes sense that the microbes would be different. So hopefully we'll get that information. And then there's the ingestible capsules that we know that you swallow a capsule and you can see through the small intestine. But now they're developing capsules to collect fluid and eventually to collect tissue. I think part of the problem is not exactly knowing where the capsule is, but I know that soon there are developments that you will be able to localize with great precision where that capsule is in that part of the gut. So I think we'll get a lot more information when we are able to partner together and find out more about the microbiome in different areas of the gut and why is it important just to health in general and different diseases, not just digestive diseases.
Thank you. I think this is especially important as, right now, microbiome transplants are being used to treat conditions like C. diff infection. But some of the latest research is showing that when you transplant microbes that you usually get from the colon into these patients, you end up maybe treating the C. diff, but the rest of the gut, especially in the upper GI tract, is disrupted. So you might be predisposing people to dysbiosis at other locations if you're not really considering carefully how to these neighborhoods, if you will, of the microbiome. So I think the spatial aspect of learning about where microbes are and how they function at their particular sites is really important.
Can I make one comment about fecal microbial transplant? I think that helps the C. difficile because that's the primary driver disease, but it may not be the primary driver of inflammatory bowel disease or IBS or other diseases. It's really multifactorial, so I think it's going to be more successful in diseases where you know that's the main driver of the disease.
Right, absolutely.
Just to follow up on that, I think currently the problem we have, there's this huge gap between fecal microbial transfer, which is literally what it sounds like, and it's transplanting an entire microbiome that might not address the patient's problem, as you said. And then these sort of supplement probiotics, where it's usually a single strain. In most cases, we actually know that these probiotics will not colonize your gut, which in some cases that could be desirable, but when someone really has a disease that's due to a damaged microbiome, we'd like to know how to deliver the right microbes that can actually colonize the right location of the gut.
Right.
So that's sort of the gap that we're working on filling in now.
Yeah, and the other thing that I think is really exciting about the microbiome is that while we talk about the gut, very often, the microbes aren't just acting locally. They can have really long-distance effects on many other organs, which is why people are interested more and more in using microbiome-based diagnostics or therapies for things like liver disease or neurological disease even. And so, Greg, can you tell us a little bit more about how the microbiome sends long-range signals, and how would this information really impact, or inform new points for interception?
Sure, yeah. So, because animals had to contend with microorganisms in their environment throughout their evolution, so all animals have lots of cells with the capability to sense signals from bacteria or other microorganisms. And so most of you know this in the context of the immune system, of course. Our immune system can recognize these microorganisms. Notably, immune cells are modal, right? They move throughout the body. You can create an immunological memory against your microbiome. But the field of immunology historically focused on this in the context of pathogenesis. Now there's a recognition that bacteria and other microorganisms that live in your gut can really have profound impacts on your immune system. Sometimes even early life exposures could have long-term impacts on the development and function of your immune system. So that's sort of one sort of pathway through which you can have effects throughout your body, right? Immunomodulation. Bacteria also secrete small molecules that can be absorbed and get into the bloodstream. So hundreds, maybe thousands of metabolites, many of which we don't understand their full functions, but because these access the blood, they can go all the way to the brain, for example. And then sort of a third pathway relates more to Elaine, your own work, actually, that impacts on the nervous system. And so just as the immune system has ways of sensing and responding to microorganisms, your neurons can see them as well in the gut and perhaps transmit signals to the brain, which some of your own work has shown examples of. So.
Thank you for the plug. I also want to make a short plug for everybody here that if you do have any questions, since we've got some time left, our code is ADAM5 on the Slido app. So I'll take some as they roll in. On a similar note, Lynn, you're used to seeing patients who suffer from this kind of multi-organ symptoms and maybe disruptions in multi-organ communication. And so how is understanding of the complex biology of the microbiome in this really informing the potential of the future for improvements in clinical care?
Yeah, I definitely think that in Western medicine particularly, we tend to focus on one organ system that we think the symptoms are being derived from. But it's really important in patients to not just look at what they're reporting, but also all the other coexistent symptoms and conditions. I take care of IBS, people with digestive diseases, but many of them will have migraine, fibromyalgia, anxiety, depression, and sometimes they precede or they follow or they're at the same time. But it's important to recognize that a lot of these conditions have shared pathophysiology, and one of the players, and that could be the microbiome. It may not be the same, maybe affecting one disease, but maybe others are affecting multiple different areas of the body. But I think the most important point is to see individuals as a whole person and understand that first. And then it would be exciting, though, to know if there's microbial targeted remedies that could help not just one area of the body, but also the whole person. But I think the goal is trying to find the underlying root of the cause, and many of them share very similar factors like stress. There's partly genetic, but familial predisposition, diet. So we have to look at all those factors that I think are so important and take the time and understand that.
And on that note, seeing these questions roll in, and of course, not surprisingly, there's a lot of them that are about probiotics and diet. And so one question from the clinical side is if you're seeing probiotics as helpful for patients, existing probiotics versus the promise of new probiotics. And also, are probiotics worth taking if you're healthy already?
I thought this might be the first conference I was at where no one's going to ask me what's the best probiotic to take. The probiotic data, I know this more for GI disease. The data is really not great. It's usually a lower quality. And so in general, you look at the evidence, it's not recommended always. It's good for antibiotic-associated diarrhea. But in general, it can help certain symptoms, but I don't think that the evidence is here yet that we know exactly what to take. I think there's definitely limitations, and as I've talked to you, Elaine, you have said they haven't found the right probiotic yet, or a combination, and I do think, as Greg was mentioned, it's not going to be a single organism. It's probably going to be a consortia of live bacteria that will be helpful. It's a good question about probiotics for health. I don't know the answer to that. I don't think anyone knows the answer. I think it's fine because I'm assuming that it would have some gut health, but it's going to be, if you're healthy, it's really going to be hard to know that long term. But I do know patients who do take it when they're healthy and they actually get symptoms, GI symptoms, that would stop it. If you're taking it for a particular disease, you would usually use it for 8 weeks, but if it's not helping, you would get off of it.
I see. And Greg, there's also a comment specifically on probiotics and diet and how it interfaces with cancer. I think this really relates to one of the initiatives from our center in terms of studying the microbiome, spatial structure, and how it relates to cancers at different sites in the colon versus in the stomach. So, could
Yeah,
you
sure.
talk about that?
So there's a lot of really recent evidence showing connections between specific microorganisms within the microbiome and both and cancers, especially throughout the gastrointestinal tract, which makes sense. There's possibility that it could affect cancers throughout the body, but we'll stick to the gut. I think that's where the evidence is clear. There's organisms that are causal there. So famously, Helicobacter pylori, been proven by Barry Marshall, who won the Nobel Prize for proving it. But now in the colon, we also have examples of specific bacteria that have been linked to tumorigenesis in the colon. And I think there are more to be discovered. But what's really, I think, hopeful is while we understand how those sort of pathogenic tumor-causing organisms can induce tumor-related pathways, we also know that there are healthy bacteria that can induce sort of healthy epithelial developmental pathways, which would be the kind of cellular response that's the opposite of cancer. And so, my sort of hope is that if we can find these right organisms and perhaps deploy probiotic treatments at the right time, like after you get a colonoscopy, say you have polyps removed, you have a higher chance to get colon cancer? Can we have a probiotic intervention that's going to correct the epithelial dysplasia that caused those polyps? So I think there's a lot of potential there. The microbiome is incredibly druggable. As you said, Lynn, there could be some side effects. We don't want to be reckless with this, but usually these are reversible. And so I think the prospects of engineering the microbiome it's sort of easier to think about genetic engineering of the microbiome versus gene therapy of our own human cells, right? It's lower risk, but there's also massive potential because of the capability that these microorganisms have.
Well, thank you very much. I think, from studying the spatial organization, knowing exactly which microbes to target where, and also considering holistic treatment of patients, we're all really here for the same purpose, is taking this complex biology and trying to use it to, extend health span and lifespan. And thank you very much for joining me, and thank you for your attention.
Thank you.
Thanks.